Well, I'd like to thank the organizers for giving me an opportunity to talk. I've never been to London, actually, so don't think me so much a yokel, but the city's fabulous. And I'd also like to thank the AV team who's behind the curtains, because I've never seen anything like this, and I think it beats Coachella. So, anyways, let's get into the story. So, I'd really like to give you an overview covering two major topics. First of all, I'll be discussing how to integrate single-cell genomics for variant analysis, particularly in the context of cancer, and how we've applied that to the analysis of primary tumors, leveraging and integrating single-cell genomics with long-read sequencing. And then, as a secondary experimental analysis for phenotypic drill down, I'll show how the similar strategy can be applied in the context of base editing single-cell genomics. So integrated single-cell genomics variant analysis. The primary topic I'd like to just go over is this clonal variation, which has been at the backbone of much of what we understand regarding cancer. The distributions of mutations that we see can be quite varied across different subpopulations. But interestingly enough, also, this is nature's version of biological engineering, because cancer tends to be a very optimized system for growth, which is unfortunate for the patients, but might provide some opportunities, thinking a little bit in the future of how this can be used for certain biotech applications. Secondarily, we are all aware about CRISPR single-cell engineering, which really provides opportunities to increase the scale of phenotypic assessment of different types of variants. And I'm going to demonstrate some methods where we can do that, which really leverage long-read applications. And so by doing this, you can assess variants across many number of cells. Individual experiments that could have taken months, many plates get reduced down to a single batch. And I also think this offers opportunity, particularly when combined with selective growth advantage or other types of selection conditions, to actually use it to start engineering the context of cells. So what is enabling all of this is that we're already familiar with the fact that single-cell RNA sequencing now is a ubiquitous tool commonly used in the characterization of cancer samples and other types of tissues. But hidden underneath that data exists additional features that can expand the breadth of what you do with single-cell RNA sequencing. So we're all already familiar with how gene expression per given cell and enumerating the individual transcripts per that cell provide these kinds of cluster plots, cell types, distinguishing biological activities. But buried within the actual system itself and within the assays and within those single-cell DNA libraries, you have access to transcript isoforms and even allelic expressed variants that can be informative. And so one way of initially doing that through the two pilot studies and proof of concept experiments that I'm going to describe is basically taking advantage of existing methods, particularly using traditional short-read sequencing, but then really starting to leverage the long-read advantages that give you the ability to query entire transcripts that can be assigned to single cells, and thus pull out these other additional features that are available. Okay, so identifying cancer mutations at single-cell resolution. It's already been known that you can do this with single-cell DNA sequencing, and there are a variety of technologies that have been used or applied for that. But I think one thing about using and leveraging expressed variants is that much of you and those who are embarked on single-cell RNA-seq have this type of assays and this type of material available. So the way that works is you fundamentally sequence individual specific genes by looking at the single-cell cDNAs. From that, you can identify transcript isoforms, which represents really an unexplored, undiscovered country that's highly biologically relevant and very impactful in cancer. You can actually detect coding allelic variants in transcripts, and while this has been described quite a bit, I think that's a little bit under-recognized, under-explored area where we can truly use and leverage some of this information for understanding phenotype and genetics of individual cells. And then with these processes, again, all driven by long-read sequencing, and what I'll show is all nanopore-based, you can assign each mutation and even associate an expression phenotype across individual cells. Okay, so another very handy feature of this that we took advantage of is leveraging the nanopore adaptive sampling ability, where you can actually isolate individual sequences from specific cDNA populations and be able to enrich these transcripts out, and that provides another dimension of information that you can layer on top of what you're doing. So in this case, we leverage long, relatively extended lists of cancer genes and other drivers to be able to select these single-cell cDNAs, look at some of the isoform structures, also begin to recognize the presence of variants that can be assigned down to individual cells, individual cell types. And so the secret sauce behind this is to really take advantage of the single-cell sequencing cDNA libraries that already exist. So the conventional workflow involves traditionally a fragmentation that is an adaptation and elimination of information regarding the limitations of the read length, but instead you take advantage of the long-read sequencing to sequence throughout the entire cDNA, leveraging the cell barcode and barcode matching processes between both short and long read to provide overlays because the same cell populations are being sequenced simultaneously. And from the enumeration using the short reads, you can define gene expression features per given cell. But then taking advantage of the nanopore sequencing and the extended long reads across the transcript, you can drill down into new features that previously have not really been leveraged. So in our first iteration of developing this pipeline, we had pre-existing data from short reads, particularly Illumina. We had barcode alignments, cell gene matrix, and cell barcodes. And through a process developed by group members, including Billy Lau, who's currently in attendance, developed a way to be able to leverage the overlapping intact cDNAs from the exact same library, exact same sample, and use a Kamer matching process that allowed us to basically overlay the barcodes from both sources with very high accuracy generally. And then using other types of strategies, including alignment of the full-length cDNAs with their exon splicing and whatnot, we can merge those data sets and take advantage of those. So to do the proof of concept study, we actually did a number of controls, but because it's a little bit more exciting, I'm going to tell you about the analysis of primary cancers, where we tested this nanopore adaptive sampling to really drill down on specific cancer driver genes, the integration of the short read data that pre-existed with the new data that came from the adaptive sampling using nanopore long reads. And then excitingly enough, we took advantage to be able to identify somatic mutations that were principal drivers for these particular cancers as they occurred within patients. And then finally, I have some nice data that show that we can actually look at rearrangements and fusions. So we did have a ground truth data set based upon diagnostic tumor sequencing through a series of cancer panels, which ranged from anywhere from 161 to 529 genes, very high coverage using standard short read sequencing, derived from biopsies of the primary metastatic site. And from that, we focused explicitly on the substitution mutations, coding mutations, and rearrangements that were present as a ground truth for this initial comparison. And so what we did is we took advantage of the long read processing with here are some of the basic items and did a combination of both looking at a genotype-based assessment as well as primary variant calling using long shot from the long read data itself. And so the type of tumors we involved included both epithelial and B-cell metastatic tumors. Here's a list of the various tumor types that we did, including appendiceal cancer and a B-cell lymphoma. And we had the advantage, we actually had 2 metastatic sites available for each patient, so we can make a couple of inferences across that. So this represents the principal data. Both of these, all these tumor samples, both metastatic sites or primary metastatic sites, had already undergone short-read single-cell RNA sequencing. We had the advantage of that data. We went through this matching process based upon adaptive sampling, where we used target cancer gene lists that are shown in that particular panel that included 333, 529, and 161. These were defined based upon the diagnostic cancer panel sequencing tests that had been done. And then from that, we were able to identify a subset of reads that had both matching long read and short read data and brought that union together to create this more informative data set. And so appendiceal cancer tends to be particularly devastating, and one of the reasons is because the cellular complexity of that tumor as it borders against the colon means that its dissemination tends to be much more varied, and oftentimes the cell populations are isolated. So it's a perfect example where you can leverage single-cell RNA sequencing to better understand the underlying genomic changes. And so this represents cancer patient 1, where we have 2 sites, one located in the omentum, which is a covering of the digestive organ system, and another that existed in the small bowel. And the derivations you're seeing here come from the standard single-cell RNA-seq with the usual clustering methodologies, including notably the epithelial cell populations, which are circled. This represents the matching long-read data, including drivers that were recognized in these patients, including KRAS and GNAS, and several others that had been reported as part of the diagnostic sequencing panel. And you can see that we can cover all of the exons very easily from individual single cells using this nanopore strategy. Here represents some of the critical drivers that we knew did for substitution mutations, and what you're seeing in the final column with cells with long reads is the identification of the transcripts which had wild type versus the mutation. Now, the sensitivity tends to be a little bit lower because we're using a minion flow cell, but I anticipate as we're able to convert over to the Prometheon, we'll have a much greater expansion and higher sensitivity with this. But you can already see from this proof of concept study, we are identifying transcripts that have the particular mutation across these 4 important drivers that were within the patient. So this example is from the T1, and here's an example from the T2. Again, the recognition that we can actually do this, and it's feasible even using a relatively low flow cell output with a minion system and adaptive sampling integrated as part of the workflow. So here's another example where KRAS G12D is coming along. This is an important target because there's small molecules being used to try to inhibit this. And again, you can recognize that from all of this big gamish that occurs as part of an appendiceal carcinoma, we can recognize that there are epithelial cell types, find the individual cells that have the mutation using a very and relatively simple strategy by overlapping and leveraging the Nanopore adaptive sampling long reads of cDNAs. And so that's recognized in both sites here. The other thing you can do is you can take advantage of this information and begin to think, is there impacts from these variations, these somatic variations that somehow destabilize the transcript? And so this is something that allows you to actually test the consequences of these mutations and variants such that you can assess, well, is that actually still expressed in relatively same levels or not? And also provides you with something that might represent a real genetic ground truth, because I, for one, don't expect a KRAS G12D to show up in a lymphocyte or whatnot. And so some of the wiggle room that you see with a traditional transcription-based cell type assignments can actually be shown to be partly false because they harbor mutations which have no place within a lymphocyte or a fibroblast. So cancer 2, same type of example. Here's a demonstration of 5 important drivers with coding mutations that are pathogenic. And by taking advantage of this, we can see the long-read sequences in which we can assign specific cells with these types of mutations that occur. So proof of concept, it clearly works. You can see it in the ovary. You can also see in the appendiceal primary, where we can see the transcripts from individual cells that encode for these mutations. So again, a demonstration right now, early days, where we can actually have this genetic overlay based upon single-cell cDNA libraries that are already present in everyone's refrigerator. And so this is a relatively flexible workflow. B-cell lymphoma is a different type of derivation. It's an immune cell derived from B cells. Typically, in the past, we've used a series of immunohistochemistry markers to identify this. The use of the single-cell aspects have provided us with much more granular ways to be able to identify tumor cells. In this example, we have a patient who had 2 metastatic sites, and we had libraries from both that underwent adaptive nanopore sampling for a number of primary targets that are known to be involved in hematologenous malignancies. And so here's a list of some of the critical ones that were called. And as an example, we readily discovered these mutations, including a series of BCL2 mutations that were introduced and associated with the actual B-cell malignancy. And we could see that there are transcriptional differences between the wild type versus the presence of this particular mutation. That was explicitly due to the fact that this is rearranged locus as part of BCL2 IGH rearrangement that causes a large increase in the expression of this transcript. So that led to some really interesting observations that can only be done from this type of adaptive sampling long-read strategy. So for example, what you're seeing regarding these individual blocks represents a series of somatic mutations that are introduced through a part of a hypermutation DNA repair process explicit for the BCL2. And every single line that you're seeing across represents a single molecule that's assigned to a single cell that have been perfectly haplotyped and phased. So you can see the phasing of these somatic variants immediately. So I thought that was kind of impressive. And then here's my favorite part. You can actually identify a fusion gene and assign it to single cells. So as I noted, the BCL2 IGH rearrangement in B-cell lymphoma, particularly the Flicker type, is a highly prevalent rearrangement that oftentimes occurs in up to 70% of patients. And the data you're seeing are individual nanopore reads where the short clip represents the variegated color, and each one of these reads have been assigned to individual B-cell tumor cells. And you can see that a fusion has occurred here between the two. So this allows you to see rearrangements at the single cell resolution level. Okay, so next part. Well, you have all this information from these variety of translational studies. You recognize all these mutations, but what do you do about them? And in fact, if you really think about it, 99% of the somatic variants that we study, or even those that we call as being pathogenic, their actual biology, their phenotype, how they mechanistically contribute to cancer is practically unknown. So by leveraging CRISPR-Cas9 nuclease or CRISPR base editor process, we can take this exact same process that I demonstrated to identify the variants, but flip it over and use it as a way to actually engineer variants into individual cells and assess the phenotypic consequences of mutations. So as an example, with cell engineering on this scale, when you're starting to consider trying to recapitulate the effects of these types of variants, you can either take advantage typically of a CRISPR deletion that can be used to knock out, say, an exon-intron splice site, or you can take advantage of base editing processes to introduce substitution mutations down to individual amino acids. And so that's practically what we did. Now, taken length, that what we did in particular is we leveraged the fact that we could use nanopore adaptive sampling or other types of traditional molecular processes to be able to identify the exact place where we'd introduced the variants. We're not using a guide RNA barcode. We're finding the edit as it's occurring within an actual transcript. And so the process has a lot of similarities. You can take a cell lines, organoids, whatever, introduce your CRISPR library, including with ribonuclear protein delivery. It's probably the only technology where you can do this because guide RNA barcoding doesn't work so well in that. You can use edited cells, do single cell preparation, and then use this modified workflow where you take advantage of both the short read transcript enumeration and the long read full transcript sequence information to identify where the edit has actually been introduced and use that to assign the affected cells. And so in our first experiments where we did this and published in Genome Biology in 2022, we took examples where we went in and we used CRISPR to knock out a series of exon-intron junctions, and we could actually study the change in transcript isoforms. So this is an example of RAC1 where we did this. The individual red lines that point to an exon are where we designed guide RNAs to knock those out. And what you're seeing on the other side, the right-hand side panel, represents the transcript isoform changes that are shown. All of the cells individually are listed as rows, and the individual guide RNAs, because this is a multiplexed experiment where all of this was done in a single culture, in a single tube, they're all recognized immediately, and so you can see that you can do this on a multiplex scale where you're introducing novel isoforms intentionally to see what they do. Taking this one point further, we've actually introduced the idea of using single-cell point mutation engineering to do the same thing, where we could directly introduce variants that lead to substitutions. Very similar strategy, no major changes here. Highly multiplexed experiments. You don't have to torture a postdoc or graduate student to do a bunch of individual cultures. It's just one culture, boom, off you go. You take advantage of transcript enumeration from single cells to identify phenotype, pathway activity, and then simultaneously merge in the long-read transcript-based genotyping to be able to determine which cells has which transcript. And so as a proof of concept, we chose several hundred p53 mutations, developed guide RNAs that were compatible with base editor technology. And it turns out actually with that, even with base editors, there's some wiggle room where a given base editor will introduce multiple substitutions, found the exact one that was intentionally designed, and all of these mutations were called from ones that have been reported within the literature. But the vast majority of these p53 mutations from tumors, there's no functional data whatsoever. So we went through this process in a single multiplexed experiment, introduced literally over 100 of these p53 mutations, assigned them to individual cells, and then was able to take advantage of the short read information to identify what the phenotype was based on pathway activity and transcriptional features. And so just to give you a hint of this, here's an example where several of these mutations were associated with the wild-type p53 state within this experiment. And most of these had not been described, and there's actually been contradictions where some of the ones we identified have been thought not to have any major pathogenic, are thought to have pathogenic effects. And then in parallel, we identified all the functionally significant ones based upon aspects regarding their transcriptional properties, pathway activity, and can clearly separate these out. So this gives us the ability to phenotype mutations individually by combining these technologies and do this on potentially a massive scale. And so just to close this out a bit, I'm going to show a little bit of non-single cell, because the same thing can actually be done on spatial-based cDNAs as well. And so using long-read sequencing, this is an example where we're using nanopore sampling from a particular colorectal cancer case, as I'm showing right here, where we can use nanopore long reads to make similar types of assignments, but then in parallel, we're also to have isoform information and even begin to detect variants that occur that are part of transcript-expressed variants that are present within these individual sectors. So in summary, I've shown a multi-omic single-cell cancer analysis, but I think hopefully I've convinced you that there's actually applications across many types of different fields of biology. In this proof of concept early stage development using a relatively low capacity adaptive sampling flow cell, we showed that it is feasible to identify cancer mutations and rearrangements from single cell RNA-seq with single cell resolution. And this provides a ready way to integrate this type of genotypic isoform information with the current single cell RNA-seq data that you may have from conventional sequencing strategies. Finally, for future improvements, we're using to study cancer variation. We're developing new technology to even boost deeper coverage so we can do this more sensitively, and taking advantage, hopefully in the near future, where we can use adaptive sampling from the higher capacity flow cells to really boost the output, increase the number of reads that we get per individual cell, apply similar methodologies to spatial analysis, and begin to make mutation-based assignments looking at expressed transcript mutations. And then finally, cancer, in its unfortunate way, represents evolution that's being used to design a perfect cell that can run amok. But I also feel like these kinds of strategies, combining this type of information, allow us to think about cellular engineering applications that could be broadly disseminated and have application across many different topics. So here are the people who do the real work. Susan Grimes, who was critical for the adaptive sampling study, Billy Lau, who's also here, and our nanopore guru, who was an inspiration for most of these studies and helped us design many of these experiments, and Hyun-Suk Kim, who's our expert CRISPR base editor person who just recently took a faculty position. My funding sources, and that's the end of the talk. Thank you very much for your attention. Appreciate it.